Research in the Bettaieb Lab is focused on identifying and characterizing novel diet, pharmacological, and genetic-based approaches to alleviate and prevent obesity, diabetes, and their associated metabolic disorders. This is achieved through the use of dietary supplements to activate metabolic pathways similar to those that exercise and caloric restriction stimulate in our body. Using both in vitro and in vivo approaches, we are able to characterize the function and contribution of key metabolic proteins and enzymes in the pathogenesis of obesity and diabetes. Our overall goal is to identify novel strategies to either alleviate or treat the diseases using pharmacological, genetic, or nutritional approaches. Some of the current projects focus on how to prevent obesity through promoting the browning of white adipose tissue and increasing energy expenditure using genetic and diet-based approaches. Recent studies suggest exploring the possibility of activating brown adipose tissue as a potential anti-obesity strategy. However, understanding the mechanisms underlying the processes of brown adipogenesis and browning is foundational for effectively promoting the strategy as a primary or secondary preventative measure against obesity. Therefore, some of our experiments are focused on characterizing novel approaches to promote the browning of white fat using natural bioactive compounds that are as efficient, or in some cases even more efficient, in promoting the transdifferentiation of white adipose cells into functional brown-like adipocytes. Hearing loss affects all age, racial, ethnic, and socioeconomic groups, with an estimated 30 million Americans having hearing loss. Age-related hearing loss, known as presbycusis, is the most prevalent type of sensory neurohearing loss and can be attributed to factors such as genetics, oxidative stress, inflammation, cochlear vascular changes, noise exposure, and autotoxic drugs. Emerging evidence indicates that phytochemicals exhibit antioxidative, anti-inflammatory, and neuroprotective properties. Phytochemicals refer to a broad class of bioactive molecules from plants that have robust effects on cell signaling. One of the research projects in the Bettaieb Laboratory is investigating the role of select phytochemicals in alleviating oxidative stress-induced damage to the cochlear cells, which are sensory cells of the auditory system, and how we could prevent hearing loss through the use of dietary supplementation with these bioactive compounds. For more information, please check our lab website at